have to introduce myself. I am based at the University of Auckland, and my research has spanned a whole range of bacteria and eukaryotes. I'm actually a cancer researcher, but I've dabbled a lot with prokaryotes. And my role today is to set the scene. So I have the job of explaining to you the most basic thing of what is an extracellular vesicle. So my title is EV, EP, or ET, Nomenclature and Reporting. You'll see where the ET comes. So if we go to the ISEV guidelines, and what I've done is added the QR code there for you so you can have a nice read of that during your extended lunch break. So the definition of an EV according to the ISEV2023 guidelines is the term extracellular vesicles refers to particles that are released from cells, are delimited by a lipid bilayer, they cannot replicate on their own, i.e. they do not contain a functional nucleus. It kind of gets a little bit more complicated when we think about what is not an EV. And so the definition of what is not an EV was nicely highlighted in this summary diagram in ISEV2023 guidelines, which kind of showed that there is this umbrella term of extracellular particles, or EPs. This is where our EPs come in. And under this, you have the vesicular and the non-vesicular forms. Under the vesicular, we see our friends, the EVs, the true extracellular vesicles. But we also have a range of other vesicle types that can be formed synthetically or as mimetics, so EV mimetics, SVs, synthetic vesicles, ACDVs, artificial cell-derived vesicles formed from cell disruption. And then we have, on the flip side, on the other side of the arm, on this family tree, underneath, still extracellular particles, we have these non-vesicular NVEPs. There's going to be a test on this later, everybody, so I hope you're taking notes. So under the NEVP category, we have all of the non-EV particles. These are made from, derived from cells, and they can contain a molecular cargo, so proteins, nucleic acids. They can contain lipids, but the key thing is they do not have that delimited membrane. So this can include, you may have heard of exomeres and supermeres. We also have lipoproteins, chylomicrons. In different sample types, we see lots of different types of these non-vesicular extracellular particles. One of the challenges that we're going to hear throughout the day is that these NVEPs and the EVs have very much overlapping physicochemical properties. And so they can have the same size, the same density, the features that we actually use to isolate our EVs. The other challenge, and we're going to hear more about this later, is that the NVEPs, I wish there was a catchier name, the NVEPs are greatly outnumbering EVs often in biological matrices such as biofluids. So what we find, and this will be highlighted in a couple of talks, is that we see lots of extracellular particle separation methods resulting in co-isolation of NVEPs and EVs. We also want to highlight in the ISEV guidelines that there is a real challenge with characterizing EVs. Many of the methods that we use to characterize, to count Western blots, anything EM and so on, also able to analyze and count and pick up these extracellular particles. And so it can be really challenging to separate between the two. And as I say, we're going to hear a lot more about this during the rest of the day. And the recommendations from the ISEV guidelines is that if you have no evidence that you have EVs in your prep, that they recommend that you work upwards and would use the generic term of EPs, these extracellular particles. So one of the challenges we've got is EV heterogeneity. And please, anybody, put your hand up if you've seen this diagram in a review article on EVs. One? I would imagine that most people, have seen this diagram, and you see the statement mammalian, I've put the brackets, mammalian extracellular vesicles are classified into 3 groups, typically based on their size and biogenesis. There are the exosomes, the microvesicles, and the apoptotic bodies. We also have this sort of simplistic view in prokaryotic work, where often gram-negative bacterial vesicles are denoted as outer membrane vesicles. The reality is much more complicated. And so one of my favorite diagrams from the last couple of years is this from ISEV President Edith Bouzas, showing the complexity of EV subtypes that are released by both mammalian cells, another review from the types of EV subtypes that are released from gram-negative bacteria. So what we have is a huge range of heterogeneity of EV production, much more complicated than that simplistic view of exosomes, microvesicles, and apoptotic bodies. So because of this heterogeneity, MySev recommends with the terminology that the use of operational terms, so for example, based on size, in 2018 in MySev, they recommended the use of small and large EVs, and that's kind of led to a little bit of confusion in the literature because one person's large EV is not another person's large EV, and it's very dependent on the isolation method. So the use can be recommended, but caution is required, and accurate reporting of methodology is needed to ensure that people actually understand what is in that EV prep. The use of the terms exosome, so coming from the multivesicular body, and ectosome, coming from the surface of the cell, are discouraged unless subcellular localization can be demonstrated. And also the terms of microvesicles and exosome-like vesicles, which can mean anything, is also discouraged in the new MySev 2023 guidelines. So we've got a real challenge with heterogeneity, and I wanted to summarize this kind of heterogeneity issue with this rather complicated diagram, but I will take you through it. So what we've got at the top is exosomes. If we simply take exosomes released from a single cell, within those exosomes, we already have a level of heterogeneity. They may be similar size, they may have very similar biophysical properties, but we know from research that they can have different surface patterns of tetraspanins, for example. So we already have a layer of heterogeneity just by looking solely at the exosome subset of EVs. We can then take a single cell. Well, I wish we could. We could take a single cell and we can look at the types of EVs that are released. These could be formed through different biogenesis mechanisms. So you start to get a bit more of a complexity in the types of EVs that are being released. So these might vary by size, by cargo, by biogenesis mechanism. And at this point, highlighted in little stars, these are the bad things. These are the things that we do not want. These are these EPs, these co-isolated extracellular particles. Then we have monoculture, and many people will be doing cell cultures. They will be using single cell lines, adherent or suspension, bacterial lines, amoebae, and so on. And these, if you look just at the picture, the variability of the states of cells within even a monoculture can really vary. So you get another layer of complexity in both not only the particles that are produced, the EVs that are produced, but also the contaminants. Then another layer of complexity when we start to think about biofluids. And this can include phloem from plants, this can include fluid from C. elegans, this can include anything, but we often will think about mammalian biofluids such as blood or urine. And what we have in there is a complexity of the cells of origin, huge number of cells of origin. We also have the complexity of cells in very different states, different growth conditions. And so again, another layer of complexity to the vesicles and the extracellular particles that are released. And then finally, this is the challenge. I think this would be really interesting to start fishing, literally, in seawater and in river water, in environmental samples, soil samples, the complexity, the diversity of the organisms that have donated to those EVs that have been released, but also those contaminants. We start to have this much more increased heterogeneity in the EVs in our populations. So please bear this in mind when you're doing your EV research. And so what this leads to is that finding rare EVs, we need a highly selective isolation method. We also may need a highly sensitive method for detection of their assay for our EVs of interest. And as you'll hear, isolation methods are unfortunately largely unable to separate this heterogeneity. But I'm really hoping by the end of the day, we're going to have some new technologies that are coming through that can help us to delineate and this crazy heterogeneity with the EVs. The other complexity that I want to highlight, and you'll hear more about through the day, is this idea of nature versus nurture. And I'm a geneticist, so I always think about nature versus nurture, and the idea of what is an EV born with and what does it acquire from its environment. So the environmental factors, for example, we had it in our quiz, the EV corona. And so EVs can mop up their environment. They're very sticky little things, and they can represent their environment by this co-isolation with a EV corona that adheres to the surface of the EVs. Now, I think of also these kind of words of co-isolates and contaminants. A contaminant for me, I think of as a weed. I give you a story. I found lots of passion fruit growing in my garden. And there were passion fruit growing up a fence, and I was like, great, and we had a really good feed from those. So we had a feed, but we also had them everywhere. And I had to spend hours digging these passion fruit that I did not want out my garden. So these were the weeds. And so we've got to think about contaminants as, are they good, or are they just these molecular weeds as a contaminant? We also then have this idea of a co-isolate. And I always try and think about, is that co-isolate isolated with the EVs or on the EVs? And I think this is really different, and it's going to change what you might isolate, how you might do the isolation. There may be some co-isolates that are just impossible to separate from your EVs because they're part of this surface corona. Also, the idea of the feed or the weed, something good or something that you don't want, may actually be inherently important to the function of the EVs. They may be involved in the targeting, they may be involved in therapeutic function, cloaking for protection from the immune system, or unfortunately, there are many papers showing that these coronas, these co-isolates, can also mask the true effects of the EVs. So, how do we report our work accurately to enable others to interpret what the EVs actually are? So, we want to avoid being the little devil here, and we want to try and be the angel. And the angel will use a study design that thinks about confounders, that will keep suitable controls. They will put best efforts, and it's always best efforts because we have limitations in publication lengths, but best efforts in reporting all methods within the manuscript. Or we also recommend using the EV track database that I've also added in this QR code. Also consideration of using multiple approaches for characterization, and also a high-level correct use of nomenclature. So use EPs if you can't prove that the EV and that membrane is actually present. So where to from here? What's on the horizon? The nomenclature, I think, is going to be constantly evolving. We saw that from the change from 2018 to 2023 with the drop of the small and large EVs. But this hopefully will simplify things, remove ambiguity, and improve biological accuracy, and also improve the quality of the publications that we're all making. I think the new guidelines will also be guided by new technologies. So while the new technologies come through, we're going to learn more about the heterogeneity, about different subsets. And I think these technological advances are going to be really beneficial. But just remember, the takeaway is, if it's an EV, make sure it has a lipid membrane. So my last comment is, you know I said EVs, EPs, and ETs? What's an ET? Well, maybe it's one of these. And I've heard that the functional transfer is making those cells phone home. So I'll leave it there. As I've said, if there's any questions for myself or the rest of the panelists, please save those, pop those onto Slido, and we'll be able to pick them up during the panel session.